Hallo. Hello, everybody. My name is Gabriele Saretzki, and I'm sharing with you today our results on the influence of dietary restriction on mouse adipocytes, and also on a model of cultured human adipocytes where we found that oleic acid is able to alleviate senescence. So just for in a matter of introduction, so here is a schematic representation of adipose tissue where you see in yellow the adipocytes, but there are of course also other cell types present, macrophages, which are recruited to it upon inflammation. There are blood vessels and there are T cells, and all of that is also, of course, connected to systemic circulation. So, as you might know, there are different types of adipose tissues: brown adipose tissue, which is mainly responsible for thermogenesis. I will not target upon that. And there's subcutaneous fat and visceral fat. And it is known that in obesity, there are larger white adipocytes, there's hyperlipidemia and dyslipidemia, lipotoxicity, as well as chronic systemic inflammation. There is glucose resistance and insulin resistance and glucose intolerance. And all these features can also lead to a metabolic syndrome. During aging, there is known that there occurs a fat redistribution to other tissues. So, for example, there is hepatic steatosis, there's also inflammation and senescence, and as in obesity, there is insulin resistance and glucose intolerance. So our initial question was, how much similarity is there between processes of aging and obesity, mainly in visceral adipose tissue? So for that, we have analyzed senescence and or aging markers. So we looked for telomeres, which are shown here as pink dots at the end of the blue chromosomes. These are mainly shortening and dividing cells, and they lead to a reversible cell cycle arrest and eventually to replicative senescence. The very good senescence marker is also the DNA damage marker gamma H2X, which we use. Other people use 53BP1. We also analyzed cell cycle checkpoint related kinase inhibitors like P21 and P16. And we looked into also, it is known that there is a pro-inflammatory phenotype connected to senescence, and we analyzed some of those on the expression level. So in addition to replicative senescence, there's also stress-induced senescence. So I might have forgot to mention that the adipocytes themselves, when they are differentiated, are mainly postmitotic, but all other features like DNA damage, cell cycle checkpoints, and the SASP also occur in non-replicative senescence, where telomeres don't shorten because the cells don't divide, but they still get damaged. And I will also show some results of that. Yeah, so other postmitotic cells are neurons, hepatocytes, muscle cells to a largest extent. Oh, sorry. So our hypothesis for the in vivo part was that aging promotes hypertrophy and senescence in mouse visceral adipose tissue. And we also switched from dietary restriction, or initially switching from normal ad libitum feeding to dietary restriction can ameliorate hypertrophy and senescence and or aging markers. And we also performed a switchback from dietary restriction to ad libitum fed in order to see whether there is a kind of memory effect. So our model, dietary restriction, for most of the aging researchers, they know that this is the only non-genetic intervention that increases longevity and health markers in various organisms, starting from primitive single-celled ones as yeast, over flies, rodents, and to rhesus macaques, and some data there might be also on humans, but it is very hard to keep this regime for a longer time. So we discriminate between short and long-term caloric restriction, and restriction itself can be in the range of 20 to 40%. And this results in a lower body weight, and you can see this here in the scheme, and of course reduced fat mass because there is mainly lean mass. So normally we start at 3 months of age, and you see in parallel there is quite a number of different difference in body weight. So our first study is a late onset, so we didn't start at 3 months, but we started around 12 months, and I show this here, or even at 14 months, and it was only for 2.5 months. So people like me who are already older can still do it if they like. And we also followed mice until the age of 30 months, which is quite old. So the second study I will talk on is long-term DR. So here indeed we started at 3 months. And yeah, I forgot to mention that in the first study, the late onset was only 26% food restricted, while the second study, which indeed started at 3 months, it was 40%. And it continued over more than a year. So you see here 12 months, 15 months, the same for DR. And at 12 months, there was the switch I mentioned before from DR to AL and from AL to DR. And this switch is essentially very similar to the first study where we switched at 14 months. So the analysis we performed was mainly adipocyte size, DNA damage, macrophage infiltration, expression of the cell cycle inhibitor kinases P16, P21, as well as pro-inflammatory marker expression. So here you can see the late onset study. You might appreciate that at 17 and a half months of AL, there was so yeah, I got a bit confused, so maybe even 17 months and not 14 months. But it doesn't matter. So here you see the adipocytes are indeed larger, while in the DR, and you can see this also here on the graph, it is indistinguishable from the 5 months young ones at 24 months. they don't increase more, and at 30 months they even shrink. And this is both analyzed by area and perimeter. So for the DNA damage and senescence markers, here you can see the same time points. And again, there is an increase at the ad libitum fat for mice which are older, so roughly 1.5 years. And however, the DR indeed decreases this DNA damage, while even higher ages have a small but continuous rise. So the number of damaged cells, so this was the number of DNA damaged foci per cell. These are the number of cells which have damage, but it is very similar. With age and increases the number. The dietary restriction, even during 2.5 months, decreases it to the level of 5 months. There's a further increase in 24 months, but at 30 months there's a decrease. So for the P16, we saw an increase, which however was only significant at high ages. However, you see that there's also a quite substantial decrease under DR, so there's less senescence. It is also quite significant. The stress marker P21 is decreased with short-term late onset dietary restriction, while at 24 months it's still high, but again it decreases at 30 months. So that means it's really, really high age for a mouse. So comparable maybe for 90 to 100-year-old person. Inflammation markers are not very consistent, but you can see from young to old there is an increase in TNF-alpha. There's a decrease of DR, however it's not significant because of the large deviation. There is a significant decrease during DR for interleukin-6, and interleukin-1 beta is not. It has a trend, but and also for the decrease, but it's not statistically significant. So we also looked for macrophage infiltration using the marker CD68 as a positive control. And when we look for numbers, they increase. However, interestingly, they don't decrease with dietary restriction. But if we look for aggregates, which I believe are shown here, there is a significant increase at 30 months. So it is known that aging is to a large extent connected to inflammation, and we think this is a readout for it. And I showed you in the initial scheme that there are macrophages coming to be infiltrating into the adipose tissue. So the second study now, if you compare AL and DR, you see again a significant decrease of the adipocyte perimeter and the area. However, if you now switch from AL to DR, this is similar to the late onset DR I just showed you. They still decrease. However, from DR to DL, there is not a major increase, and this is shown here. 12 months ad libitum, 12 months DR, 15 months AL, 15 months DR, and then the AL to DR, where it still decreases significantly, but the DR to AL, it keeps the small size. So there is a clear memory effect. For the DNA damage, the picture is very similar. It is high at ad libitum, it decreases with DR, and it decreases again with a late onset switch from AL to DR. But if we now go back to AL from DR, again, the memory effect keeps in. So for the macrophage aggregates, there is a significant DR effect. However, there's no effect, and I think that is similar to what we showed in the first study. Apparently, the time is not enough for short-term 3 months switch from 15 months, so at 12 months they were switched to DR. However, if we switch back from the dietary restriction to AL, there is still a decrease. So again, a memory effect. So the conclusions from this aging in DR in vivo in visceral adipose tissue is that aging in the mouse VAT is indeed associated with cell senescence. It increases hypertrophy and both parameters coincide, and as senescence we had the DNA damage and the P16 and P21. The dietary restriction, either short or long-term, early as well as late onset, are able to protect against AL-induced senescence markers, except for the macrophage aggregates. And importantly, there is a metabolic memory for the dietary restriction for several, but not all, senescence-related parameters. And this memory effect of the DR is in good alignment with previously shown experiments on the same set of mice, where it was also shown that glucose tolerance increases and insulin sensitivity keeps going. That means insulin insensitivity is less pronounced. So in the second set of experiments, I already mentioned that before, we used primary human adipocytes. We differentiated them both subcutaneous and omental, and we used palmitic acid, which is a saturated fatty acid, in order to model stress conditions. And our question was, does that result in senescence? And then we treated these differentiated and stressed with palmitic acid adipocytes with oleic acid, which is a healthy unsaturated fatty acid, mainly known from the Mediterranean diet. And we also looked for effects there. So again, or interestingly, or quite we were quite happy to find that the palmitic acid indeed induces hyperoxia. Oh, sorry, I didn't mean to change it. I just wanted to get rid of that. Hypertrophy in subcutaneous cells, and we treated them at the same time. Oh, sorry, I just try to get rid of this, but so here you see the untreated differentiated adipocytes. These are treated with OA and with PA, and we looked at different time points. In OA, nothing changes, but with PA, it increases from 2 to 4 days of treatment, and of course, compared to the non-treated. So these were commercially available human adipocytes from Lonza, but we had also donor-derived one, where we had from the same donor subcutaneous and omental ones, and the subcutaneous responded to the palmitic acid the same way as the Lonza ones. So we had here the deviation is from different donors, while here the experiments were just repeated with the same cells. And there is a trend also in the omental one, but omental ones are a bit different. So we mainly don't see such a highly, such a good effect, but mainly just a trend. This might be due to the slower growth of omental adipocytes initially. How do I get back of this here? Anyway, so when we look for H2X, and you can see these green dots here on a cell nucleus, and the red is the P16 staining, we can show that the untreated and the OA treated don't have a lot of H2X, while the palmitic acid indeed shows a lot of DNA damage. And we also looked here for telomere-associated DNA foci, where we have the DNA damage in green and the telomere signal in red, and we look for those which overlap and show yellow signals. and it is known that telomeres are very sensitive against oxidative damage and also to senescence induced by PA. And while the increase here is fourfold, here you can see we have hardly any telomere-induced or telomere-associated foci, but with PA there is more than 40-fold increase. So, and also for the P16, here shown in the red signal, it is very similar to what we found for the DNA damage. P16 is rather late senescence marker, and again, OA doesn't show any increase, but PA does. So, we then started to do some co-treatments where we either switched from palmitic acid to oleic. We have only the oleic acid treatment, which is essentially what I showed you before. We have oleic acid to palmitic acid, or we have palmitic acid and oleic acid together. And yeah, after 3 days, after 2 days and after 4 days. So this is what we found before, only the PA has an influence. Again, the same here, only PA. If you shift the PA to OA, there's no major effect. The OA is low, and the PA to OA shows an immediate effect, but nothing statistically significant. But we find a significant effect on the DNA damage. On the DNA damage. So again, we show here the subcutaneous and the omental, and also for the P16. And if we do now the co-cultivation, we see that the shift from oleic to PA increases. Let me see. So if we shift from PA to OA, it stays the same as it did for the size. OA is low, but if we co-culture, or if we shift from OA to PA, it increases. So palmitic acid seems to have a kind of dominant effect. But if we now do the same co-culture of these adipocytes with OA and with PA, we see a significant difference. So the OA is able to ameliorate the effect of the palmitic acid on its own. There is a trend for P16, but unfortunately it's not significant. So the conclusions from that is that PA treatment in vitro indeed induces hypertrophy in senescence. So that means indeed this treatment is a good model for senescence. And again, we use these markers, DNA damage and P16. The hypertrophy correlates with the DNA damage and the high P16. And OA is able to counteract the PA-induced DNA damage only when it's used in co-treatment, not if it's shifted after 2 days and cultured further 2 days. And that means there are indeed a lot of shared pathways between aging and obesity, and these might even co-occur and exacerbate each other. So this is just a scheme, what we did. We looked for aging, for treatment of cultured adipocytes with palmitic acid, and there's also metabolic stress. All of these increase hypertrophy, DNA damage, P16 is senescence, inflammation, macrophage numbers, and the interventions which are able to ameliorate these shown effects are dietary restriction, oleic acid, and we also did some study in collaboration with Moroccan scientists, but I didn't have the time to show it here. So the person who conducted most of these experiments are my then PhD student Abbas Ishak, other master students, undergraduate, international students were involved. Also, our nutritionist and my collaborator, Professor von Sklinitzky in Cameroon. And for the human adipocytes, we were lucky or happy to receive them from the Mayo Clinic, from Professor James Kirkland and Tamara Shkonia. And that's all. And yeah, we'll see whether there are any questions.